Hello student, welcome in today's session. Today we are going to learn about the second experiment from this chapter, that is the molecular basis of inheritance, and we start with the, in last video we start with the experiment that is known as a Griffith experiment, and these all experiments are about what is the genetic material in the organism, that is the DNA, and to confirm that genetic material is DNA, that experiments are carried out. The first experiment is Griffith experiment, which is carried out in 1928. After the 16 years of that experiment and after the 10 years of hard working, that three scientists, that Oswald Avery, then Macleod and Macleod-McCarty, they carried out one of the experiment on the DNA, RNA, protein and other material like the carbohydrate from that heat killed S type plus R type, and that experiment it is known as Avery-Macleod and McCarty experiment. Today we are going to learn about this experiment and Hershey and Chase experiment. These two experiments today we are going to learn. That in 1944, after the 10 years of their hard working, they are from the Rockefeller University. Then they purified them. That you know that after a Griffith experiment, Griffith extracted blood sample from the mice that were killed, that were no more. Then that is the heat killed S type plus R type, the mixture of heat killed S type and R type. That blood sample it is collected by Griffith and he carried out his experiment. But these three scientists also carried out experiment from the blood samples of that heat killed S type and R type. That something is a transform from that heat killed S type to R type, and therefore R type become S type. R type is a non-virulent and it is become S type, that is the virulent. It causes diseases, it become pathogenic. And why it is become pathogenic? Because something is transferred from heat killed S type to R type to make it S type, and that something is called as transforming principle. असं काहीतरी होतं जे हीट किल्ड एस टाईपमधून आर टाईपला ट्रान्सफर व्हायचं, ज्यामुळं आर टाईप हा एस टाईप होऊ लागला होता. नॅचरली आर टाईप हा रोग कॉज करत नाही, तो नॉन व्हायरुलंट आहे, पण तो जेव्हा एस टाईपचा झाला तेव्हा तो रोग कॉज करू लागला आणि न्यूमोनिया होऊन माईस मरू लागले. दॅट बॅक्टेरिया वॉज द डिप्लोकॉकस निमोनी, स्ट्रेप्टोकॉकस निमोनी. आणि जी प्रोटीन कोड डेव्हलप करण्याची जी नॅचरल जी प्रोसेस आहे ती कशी होते बॅक्टेरियामध्ये ते शोधण्यासाठी ग्रिफिथचं एक्सपेरिमेंट होतं. अँड दॅट समथिंग इट वॉज नथिंग बट द ट्रान्सफॉर्मिंग प्रिन्सिपल अँड दॅट वेअर कन्क्लुडेड ॲज अ DNA. For that purpose, that these three scientists, Avery, Macleod and McCarty, they carried out experiment. Firstly, what they're doing, they purified the DNA, RNA, protein and other material like the carbohydrate. That means they purified the DNA, RNA, protein and carbohydrate from the mixture of heat killed S type and R type. They carried out four samples from that, four extraction from that mixture. Then what they do, that after that they find out that they given that heat killed, they transformed, they purified the DNA, RNA, protein and other material from heat killed S type, and they given that material to R type. They give that material to R type of bacteria, and after that they were found that RNA is not transforming, protein it was not transforming, carbohydrate it was not transforming, but DNA only transformed from heat killed S type to R type. They purified the four extraction from that heat killed S type and they given that extraction to R type. What is exactly transferred from heat killed S type to R type? Only DNA is getting transformed from heat killed S type to R type, and R type become virulent, that is the plain S type. Then only DNA is found to be transforming principle from S strain to R strain. हीट किल्ड S strain पासून कोण ट्रान्सफर होत होता? R टाईपमध्ये फक्त DNA ट्रान्सफॉर्म होत होता. आणि हे कशावरून लक्षात आलं? त्यांनी RNAs, प्रोटीन ब्रेकडाऊन करणारे एन्झाईम, कार्बोहायड्रेट ब्रेकडाऊन करणारे एन्झाईम्स वापरून बघितले तर कुठलंच मटेरियल ब्रेकडाऊन होत नव्हतं आणि पुढे R टाईपच्या याच्यामध्ये ट्रान्सफर होत नव्हतं. बट वेन these three scientists used the DNA nucleases, that is the endonucleases type of enzymes which broke down the DNA. When they mix the DNA with the DNA nucleases, there were no transformation. बघा, त्यांनी RNA प्रोटीन तोडणारे एन्झाईम वापरून बघितले, त्याचा काही फरक पडला नाही. बट वेन दे यूज द DNA breakdown enzymes, the enzymes which are breaking the DNA, that time transformation could not take place. That means only DNA, it was transferred from heat killed S type to R type. And instead of mice, organisms, animals, they use the test tube assay method. They use the heat killed S type and R type mixture in a test tube setup. Therefore, it is called as test tube assay method. Then that when they using the DNA nucleases enzyme, that time transformation doesn't take place. That means DNA get damaged by that enzyme. And when this enzyme only affected on the DNA, that means DNA, it is the material which is transferred from heat killed S type to R type. And this experiment proves that transforming principle was DNA, but not all biologists were not convinced with their experiment. हे या 3 सायंटिस्टचं एक्सपेरिमेंट होतं जे ग्रिफिथ एक्सपेरिमेंटला पुष्टी देत होतं, त्याला सपोर्ट करत होतं. त्याच्या 16 वर्षाच्या कालावधीनंतर, ग्रिफिथच्या कामाच्या 16 वर्षाच्या कालावधीनंतर दॅट रॉकफेलर युनिव्हर्सिटीचे ह्या 3 सायंटिस्टनी काय केलं? त्यांनी हीट किल्ड एस टाईपचे DNA, RNA, प्रोटीन आणि कार्बोहायड्रेट सेपरेट केले आणि ते R टाईपला इंजेक्ट करून बघितले. आणि त्यांनी बघितलं की काय ट्रान्सफर होतं हीट किल्ड एस टाईपमधून R टाईप. There were no RNA, no proteins, no other material, but only DNA is transforming. And when they're using the DNA breakdown enzymes, that time transformation doesn't take place. This clearly proves that the DNA, it is only the transforming principle which transforms from heat killed S type to R type. But all biologists were not convinced with their experiment. Therefore, the next experiment carried out by Alfred Hershey and Martha Chase, and that they carry this experiment, it is known as the Hershey and Chase experiment, and they worked over the viruses which are infecting the bacteria, and that is called as the viruses which are infecting the bacteria, they are called as bacteriophages. Hershey and Chase carried out the experiment on the bacteriophages. Then you know that the structure of bacteria, they are having the outer protein coat. Bacteriaचं structure थोडंफार आपण बघू, that they are having the outer protein coat. रेड कलर मध्ये प्रोटीन कोट दाखवते मी. And they are having the inner genetic material. Outer protein coat, it is called as a capsid. It is nothing but the capsid. And inner genetic material, it is made up of that DNA. And you know that DNA is composed of a phosphorus and proteins are composed of a sulfur. That means that phages or bacteriophages, they are composed of DNA and protein. Outer covering made up of protein and inner genetic material, they are made up of DNA. When any bacteriophage, when any bacterial viruses, they are infecting the bacteria, they put up only their genetic material inside that bacterial cell. The outer covering remain outside, that is called as ghost. And when the bacteriophages are allowed to infecting on the bacteria, and after centrifugation, we will observe that all that capsids, protein coat, remain in the supernatant solution, supernatant liquid, which were decant by the scientist. आपण जेव्हा DNA, प्रोटीन, RNA मिळवतो तेव्हा वरचं पातळ असलेलं सोल्युशन काढून टाकण्यात येतं, खालच्या घट्ट मटेरियलमधून आपल्याला पुन्हा दॅट मटेरियल शोधायचं असतं जे आपल्याला DNA प्रोटीन शोधायचे आहेत ते. बघा, phages composed of DNA and protein. When these phages or bacterial viruses infecting the bacteria, only genetic material entered in the host cell. फक्त काय entered होतं? Genetic material entered होतं. And when genetic material entered in that bacterial cell, that viral DNA that disturbs the protein synthesis of host cell, that makes their own protein with the help of bacterial DNA, and the synthesis again the capsid. त्यांचा पुन्हा प्रोटीन कोट आत जाऊन सिंथेसिस होईल, पण जाताना ते त्यांचा प्रोटीन कोट सोबत नेत नाहीत. देन दे वर्क फॉर इट वॉज प्रोटीन ऑर अ DNA. त्यांचं काम कशासाठी होतं? हर्शी अँड चेस एक्सपेरिमेंट. इट वॉज डन फॉर दॅट ट्रान्सफॉर्मिंग प्रिन्सिपल. इट वॉज DNA ऑर अ प्रोटीन. बट again they uses the radioactive samples. They use the radioactive protein and radioactive sulfur. They take the many bacteriophages. त्यांनी खूप सारे bacterial viruses घेतले and to synthesize their structure they uses some viruses which are made up of genetic material with the radioactive phosphorus. काही viruses, काही bacterial viruses चा DNA हा radioactive phosphorus नी बनलेला होता आणि काही bacterial viruses चा प्रोटीन कोट radioactive sulfur नी बनलेलं होतं. They uses many bacterial viruses. Hershey and Chase make some viruses with the radioactive protein. Outer protein coat it is made up of radioactive sulfur, and some viruses with the inner genetic material with radioactive phosphorus. काही bacterial viruses त्यांचा आतलं genetic material radioactive phosphorus बनलेलं होतं आणि काही याचा प्रोटीन कोट radioactive sulfur पासून बनलेला होता. Then Hershey and Chase allowed that two types of bacterial viruses infecting on the E. coli type of bacteria. आणि एका पेट्री डिशमध्ये ठेवलेल्या E. coliच्या कॉलनीजवर हे व्हायरसेस त्यांनी इन्फेक्ट करण्यासाठी ठेवले. अँड आफ्टर सम टाईम ही फाइंड आउट दॅट त्यांनी काय केलं, हे इन्फेक्शन होऊ दिलं आणि त्यांनी ते बॅक्टेरियाचं सोल्युशन, ई कोलायचं सोल्युशन काय केलं, सेंट्रिफ्युगेशन केलं, फिरवलं, जसं मिक्सरमधून सोल्युशन फिरवतो आपण एखादं. आफ्टर सेंट्रिफ्युगेशन then they observed that E. coli under microscope. Then E. coli shows radioactive phosphorus but not radioactive sulfur. बघा, they observed only the E. coli with radioactive phosphorus but no E. coli with the radioactive sulfur is there, because after centrifugation protein coats are removed which is made up of radioactive sulfur. बघा, काही बॅक्टेरियल व्हायरसेसचे प्रोटीन कोट कसे होते, प्लेन होते, काहीचे रेडिओक्टिव्ह सल्फरपासून बनलेले होते. म्हणजे प्लेन प्रोटीन कोट पण नाही आले, सल्फरनी बनलेले रेडिओक्टिव्ह प्रोटीन कोट पण नाही आले. बट सम बॅक्टेरियल व्हायरसेस दे आर हॅव्हिंग जेनेटिक मटेरियल मेड अप ऑफ रेडिओक्टिव्ह फॉस्फरस अँड जेनेटिक मटेरियल इज ट्रान्सफर्ड फ्रॉम बॅक्टेरियल व्हायरस टू बॅक्टेरिया ई कोला अँड रेडिओक्टिव्ह फॉस्फरस इज ऑब्झर्व्ड इन द DNA विच मेक्स द DNA रेडिओक्टिव्ह इन नेचर. त्यांनी दोन प्रकारचे बॅक्टेरियल व्हायरसेस घेतले होते, एकाचा रेडिओक्टिव्ह फॉस्फरस होता आणि एकाचा रेडिओक्टिव्ह सल्फर होता. फॉस्फरसनी प्रोटीन DNA बनतात आणि सल्फरनी प्रोटीन बनतात. प्रोटीन कुठं असतं, आउटसाइड कवरिंग, इट इज कॉल्ड एज प्रोटीन कोट, अँड जेनेटिक मटेरियल मेड अप ऑफ DNA विच इज मेड अप ऑफ रेडिओक्टिव्ह फॉस्फरस विच इज प्रेझेंट इनसाइड. काहीचा आतला भाग चमकायचा, रेडिओक्टिव्हिटी म्हणजे इट गेट अ फ्लोरोसन्स, काही रेज सोडतात, त्या मटेरियलला रेडिओक्टिव्ह मटेरियल म्हणतो. दॅट after centrifugation E. coli shows radioactive phosphorus, radioactive genetic material but not radioactive sulfur. That means protein were not transferred from heat killed S type to R type but DNA were transformed from heat killed S type to R type, and it indicate that ह्या एक्सपेरिमेंटनी काय इंडिकेट, इट इंडिकेट दॅट DNA entered in the bacterial cell, not the protein. That means DNA only is the genetic material which transform from one cell to the another cell, which transform from heat killed S type to R type. Again we will revise this Hershey and Chase experiment. They carried out the experiment on the bacteriophages. What are the bacteriophages? They are viruses infecting or attacking on the bacteria. What is the structure of bacteriophages? That outer protein coat and inner genetic material, that is outer capsid and inner core. Then that when viruses infect the bacteria, protein coat remain outside and only genetic material enter inside the host cell, and their work to be find out whether the genetic material is DNA or protein. And they make that all bacterial viruses radioactive with some viruses with the radioactive phosphorus, and they treat the some viruses with radioactive sulfur. काही व्हायरसेसचा आउटर प्रोटीन कोड सल्फरनी बनवला आणि काही व्हायरसेसचं आतलं जेनेटिक मटेरियल रेडिओएक्टिव्ह फॉस्फरसनी बनवलं. अँड दे अलाउ टू इनफॅक्ट दॅट ऑल व्हायरसेस ऑन द ई कोलाय. मग काय ई कोलायवर ट्रान्सफर होत होतं? काही बॅक्टेरियल व्हायरसेसकडे रेडिओएक्टिव्ह फॉस्फरस होता, काही बॅक्टेरियल व्हायरसेसकडे रेडिओएक्टिव्ह सल्फर होता. पण सल्फर हा आउटर प्रोटीन कोटमध्ये राहतो. आफ्टर इन्फेक्टिंग दीज व्हायरसेस ऑन ई कोलाय, दॅट ई कोलाय सोल्युशन गेट्स सेंट्रिफ्युज्ड इन सेंट्रिफ्युगेशन machine, and after centrifugation that sample were observed under the microscope, and Hershey-Chase find out that E. coli with the only radioactive DNA, radioactive phosphorus DNA which contain radioactive phosphorus, they do not contain the protein which are made up of radioactive sulfur. That means the transforming principle, it is the only and only DNA, not the RNA, protein, carbohydrate and other material from the cell. These two experiments, they proves that genetic material in an organism, it is the DNA, and it is the transforming principle which were transformed from heat kill S type to R type and make that R type to S type. This all for today's, and these are 3 experiments which supports the genetic material is only the DNA. In the most of the organism is DNA, 99%, 99, only 1% that are the plant viruses contain the genetic material RNA. Thank you student for today's video.